Imagine you were alive back in the 1980s and were told that computers would soon take over everything from shopping to dating and the stock market. That billions of people would be connected via a kind of web. That you would own a handheld device orders of magnitude more powerful than supercomputers. It would seem absurd, but then all of it happened. Science fiction became our reality and we don't even think about it. We're at a similar point today with genetic engineering. So let's talk about it. Where it came from, what we're doing right now, and about a recent breakthrough that will change how we live and what we perceive as normal forever. Humans have been engineering life for thousands of years. Through selective breeding, we strengthened useful traits in plants and animals. We became very good at this, but never truly understood how it worked, until we discovered the code of life, deoxyribonucleic acid, DNA, a complex molecule that guides the growth, development, function, and reproduction of everything alive. Information is encoded in the structure of the molecule. 4 nucleotides are paired and make up a code that carries instructions. Change the instructions and you change the being carrying it. As soon as DNA was discovered, people tried to tinker with it. In the 1960s, scientists bombarded plants with radiation to cause random mutations in the genetic code. The idea was to get a useful plant variation by pure chance. Sometimes it actually worked too. In the 70s, scientists inserted DNA snippets into bacteria, plants, and animals to study and modify them for research, medicine, agriculture, and for fun. The earliest genetically modified animal was born in 1974, making mice a standard tool for research, saving millions of lives. In the 80s, we got commercial. The first patent was given for a microbe engineered to absorb oil. Today, we produce many chemicals by means of engineered life, like life-saving clotting factors, growth hormones, and insulin, all things we had to harvest from the organs of animals before that. The first food modified in the lab went on sale in 1994. The Flavor Saver tomato, a tomato given a much longer shelf life via an extra gene that suppressed the buildup of a rotting enzyme. But GM food and the controversy surrounding them deserve a video of their own. In the 1990s, there was also a brief foray into human engineering. To treat maternal infertility, babies were made that carried genetic information from 3 humans, making them the first humans ever to have 3 genetic parents. Today, there are super-muscled pigs, fast-growing salmon, featherless chickens, and see-through frogs. On the fun side, we made things glow in the dark. Fluorescent zebrafish are available for as little as $10. All of this is already very impressive, but until recently, gene editing was extremely expensive, complicated, and took a long time to do. This has now changed with a revolutionary new technology now entering the stage: CRISPR. Overnight, the costs of engineering have shrunk by 99%. Instead of a year, it takes a few weeks to conduct experiments, and basically everybody with a lab can do it. It's hard to get across how big a technical revolution CRISPR is. It literally has the potential to change humanity forever. Why did this sudden revolution happen, and how does it work? Bacteria and viruses have been fighting since the dawn of life. So-called bacteriophages or phages hunt bacteria. In the ocean, phages kill 40% of them every single day. Phages do this by inserting their own genetic code into the bacteria and taking them over to use them as factories. The bacteria try to resist, but fail most of the time because their protection tools are too weak. But sometimes bacteria survive an attack. Only if they do so can they activate their most effective antivirus system. They save a part of the virus DNA in their own genetic code in a DNA archive called CRISPR. Here, it's stored safely until it's needed. When the virus attacks again, the bacterium quickly makes an RNA copy from the DNA archive and arms a secret weapon, a protein called Cas9. The protein now scans the bacterium's insides for signs of the virus invader by comparing every bit of DNA it finds to the sample from the archive. When it finds a 100% perfect match, it's activated and cuts out the virus DNA, making it useless, protecting the bacterium against the attack. What's special is that Cas9 is very precise. almost like a DNA surgeon. The revolution began when scientists figured out that the CRISPR system is programmable. You can just give it a copy of DNA you want to modify and put the system into a living cell. If the old techniques of genetic manipulation were like a map, CRISPR is like a GPS system. Aside from being precise, cheap and easy, CRISPR offers the ability to edit live cells, to switch genes on and off, and target and study particular DNA sequences. It also works for every type of cell, microorganisms, plants, animals or humans. But despite the revolution CRISPR is for science, it's still just a first-generation tool. More precise tools are already being created and used as we speak. In 2015, scientists used CRISPR to cut the HIV virus out of living cells from patients in the lab, proving that it was possible. Only about a year later, they carried out a larger-scale project with rats that had the HIV virus in basically all of their body cells. By simply injecting CRISPR into the rats' tails, they were able to remove more than 50% of the virus from cells all over the body. In a few decades, a CRISPR therapy might cure HIV. And other retroviruses, viruses that hide inside human DNA like herpes, could be eradicated this way. CRISPR could also defeat one of our worst enemies, cancer. Cancer occurs when cells refuse to die and keep multiplying while concealing themselves from the immune system. CRISPR gives us the means to edit your immune cells and make them better cancer hunters. Getting rid of cancer might eventually mean getting just a couple of injections of a few thousand of your own cells that have been engineered in the lab to heal you for good. The first clinical trial for a CRISPR cancer treatment on human patients was approved in early 2016 in the US. Not even a month later, Chinese scientists announced that they would treat lung cancer patients with immune cells modified with CRISPR in August 2016. Things are picking up pace quickly. And then there are genetic diseases. There are thousands of them, and they range from mildly annoying to deadly or entail decades of suffering. With a powerful tool like CRISPR, we may be able to end this. Over 3,000 genetic diseases are caused by a single incorrect letter in your DNA. We are already building a modified version of Cas9 that is made to change just a single letter, fixing the disease in the cell. In a decade or two, we could possibly cure thousands of diseases forever. But all of these medical applications have one thing in common. They are limited to the individual and die with them, except if you use them on reproductive cells or very early embryos. But CRISPR can and probably will be used for much more: the creation of modified humans, designer babies, and will mean gradual but irreversible changes to the human gene pool. The means to edit the genome of a human embryo already exist, though the technology is still in its early stages, but it has already been attempted twice. In 2015 and 2016, Chinese scientists experimented with human embryos and were partially successful on their second attempt. They showed the enormous challenges we still face in gene editing embryos, but also that scientists are working on solving them. This is like the computer in the 70s. There will be better computers. Regardless of your personal take on genetic engineering, it will affect you. Modified humans could alter the genome of our entire species because their engineered traits will be passed on to their children and could spread over generations, slowly modifying the whole gene pool of humanity. It will start slowly. The first designer babies will not be overly designed. It's most likely that they will be created to eliminate a deadly genetic disease running in a family. As the technology progresses and gets more refined, more and more people may argue that not using genetic modification is unethical because it condemns children to preventable suffering and death and denies them the cure. But as soon as the first engineered kid is born, a door is opened that can't be closed anymore. Early on, vanity traits will mostly be left alone. But as genetic modification becomes more accepted and our knowledge of our genetic code enhances, the temptation will grow. If you make your offspring immune to Alzheimer, why not also give them an enhanced metabolism? Why not throw in perfect eyesight? How about height or muscular structure? Full hair? How about giving your child the gift of extraordinary intelligence? Huge changes are made as a result of the personal decisions of millions of individuals that accumulate. This is a slippery slope. modified humans could become the new standard. But as engineering becomes more normal and our knowledge improves, we could solve the single biggest mortality risk factor, aging. 2/3 of the 150,000 people who die today will die of age-related causes. Currently, we think aging is caused by the accumulation of damage to our cells, like DNA breaks and the systems responsible for fixing those wearing off over time. But there are also genes that directly affect aging. A combination of genetic engineering and other therapy could stop or slow down aging, maybe even reverse it. We know from nature that there are animals immune to aging. Maybe we could even borrow a few genes for ourselves. Some scientists even think biological aging could be something that eventually just stops being a thing. We would still die at some point, but instead of doing so in hospitals at age 90, we might be able to spend a few thousand years with our loved ones. Research into this is in its infancy, and many scientists are rightly skeptical about the end of aging. The challenges are enormous, and maybe it is unachievable. But it is conceivable that people alive today might be the first to profit from effective anti-aging therapy. All we might need is for someone to convince a smart billionaire to make it their next problem to solve. On a bigger scale, we certainly could solve many problems by having a modified population. Engineered humans might be better equipped to cope with high-energy food, eliminating many diseases of civilization like obesity. In possession of a modified immune system, with a library of potential threats, we might become immune to most diseases that haunt us today. Even further into the future, we could engineer humans to be equipped for extended space travel and to cope with different conditions on other planets, which would be extremely helpful in keeping us alive in our hostile universe.
A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to defuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. The opportunity to do this kind of genome editing also raises various ethical issues that we have to consider, because this technology can be employed not only in adult cells, but also in the embryos of organisms, including our own species. When viruses infect a cell, they inject their DNA. And in a bacterium, the CRISPR system allows that DNA to be plucked out of the virus and inserted in little bits into the chromosome, the DNA of the bacterium. And these integrated bits of viral DNA get inserted at a site called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A big mouthful. You can see why we use the acronym CRISPR. It's a mechanism that allows cells to record over time the viruses that they have been exposed to. And importantly, those bits of DNA are passed on to the cell's progeny. So cells are protected from viruses not only in one generation, but over many generations of cells. This allows the cells to keep a record of infection. And as my colleague Blake Wiedenheft likes to say, the CRISPR locus is effectively a genetic vaccination card in cells. Once those bits of DNA have been inserted into the bacterial chromosome, the cell then makes a little copy of a molecule called RNA, which is orange in this picture, that is an exact replicate of the viral DNA. RNA is a chemical cousin of DNA, and it allows interaction with DNA molecules that have a matching sequence. So those little bits of RNA from the CRISPR locus they bind to protein called Cas9, which is white in the picture, and form a complex that functions like a sentinel in the cell. It searches through all of the DNA in the cell to find sites that match the sequences in the bound RNAs. And when those sites are found, as you can see here, the blue molecule is DNA. This complex associates with that DNA and allows the Cas9 cleaver to cut up the viral DNA. Makes a very precise break. So we can think of the Cas9 RNA sentinel complex like a pair of scissors that can cut DNA. It makes a double-stranded break in the DNA helix. And importantly, this complex is programmable. So it can be programmed to recognize particular DNA sequences and make a break in the DNA at that site. As I'm going to tell you now, we recognize that that activity could be harnessed for genome engineering to allow cells to make a very precise change to the DNA at the site where this break was introduced. That's sort of analogous to the way we use a word processing program to fix a typo in a document. The reason we envisioned using the CRISPR system for genome engineering is because cells have the ability to detect broken DNA and repair it. So when a plant or animal cell detects a double-stranded break in its DNA, it can fix that break either by pasting together the ends of the broken DNA with a little tiny change in the sequence at that position, or it can repair the break by integrating a new piece of DNA at the site of the cut. So if we have a way to introduce double-stranded breaks into DNA at precise places, we can trigger cells to repair those breaks by either the disruption or incorporation of new genetic information. So if we were able to program the CRISPR technology to make a break in DNA at the position at or near a mutation causing cystic fibrosis, for example, we could trigger cells to repair that mutation. So once a double-stranded break is made in DNA, we can induce repair and thereby potentially achieve astounding things like being able to correct mutations that cause sickle cell anemia or cause Huntington's disease. I actually think that the first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues. Right now, a lot of the work that's going on applies to animal models of human disease, such as mice. The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. Now, in this example, the CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color of these mice. Imagine that these white mice differ from their pigmented littermates by just a tiny change at one gene in the entire genome. And they're otherwise completely normal. And when we sequence the DNA from these animals, we find that the change in the DNA has occurred at exactly the place where we induced it using the CRISPR technology.
Let's begin by talking about how CRISPR-Cas9 works naturally in a bacteria like Streptococcus pyogenes or E. coli or something like that. Essentially, CRISPR-Cas9 in a bacteria acts as an adaptive immune response. That is, it remembers when a virus has infected the cell in the past, and it keeps a little bit of viral DNA and uses it so that if the same species of virus infects the cell again, it will be able to respond to it much more quickly and more effectively. You can see here in my diagram that over here we have a bacteriophage, that's a kind of virus that infects bacteria, injecting its DNA into this bacterial cell like a syringe. It injects its DNA into the bacterial cell. And the first thing that happens is a pair of enzymes called Cas1 and Cas2. They are actually two separate enzymes, but they function together. They're joined at the hip, a bit like thing 1 and thing 2 in the Cat in the Hat. They're always together and they work in concert. And what they will do is they will cut out a region of the virus's DNA called a protospacer and stick it into part of the bacterial chromosome that's called a CRISPR array. Now we'll talk about the CRISPR array in just a moment, but you'll notice that in the CRISPR array there are repeats separated by spacers. And so this protospacer will become a spacer in the CRISPR array. The term proto means ahead of or before, and that's exactly what happens. You know, these enzymes Cas1 and Cas2 identify this and think, well, that would be a suitable spacer. Let's turn it into a spacer. So it's a protospacer, and they make it into a spacer. Of course, they don't really think anything. They're enzymes, they don't have brains, but you get the point. Now, the spacer gets inserted at the 5' end of the CRISPR array. All right, that is at the 5' end of the complementary strand of the CRISPR array. So it gets put there, and then they build a new repeat region afterwards. And you'll notice that there's a repeat after each spacer. Spacer, repeat, spacer, repeat, spacer, repeat. Every one of those repeats is exactly the same as all the other repeats. That's why it's called a repeat. And the spacers, of course, are in between them. The term CRISPR array, or CRISPR, the word CRISPR, stands for clustered, regularly interspaced palindromic regions. And let's just break that term down a little bit. Clustered, of course, means all together because this CRISPR array is all in one place on the chromosome. Okay, all these spaces are all together in one cluster. Regularly interspaced is referring to the spaces that are regularly placed between these repeats along the CRISPR region. The palindromic repeats, well, the repeats part of that makes sense, right? Because these repeats are repeated. And the palindromic bit simply means that there are regions within those repeats that read the same on both strands in a 5' to 3' direction. Now, when it comes to Cas1 and Cas2, taking a protospacer and turning it into a spacer, they don't just cut the DNA randomly in any old place. They cut it at a precise location just upstream of a protospacer adjacent motif. Okay. Now remember, we said that scientists can use homology-directed repair to insert a foreign gene into the genome of a species. So how is that done? Well, this is what we do. We're going to create a piece of synthetic DNA. That is, you make it in a laboratory. And because we know exactly where Cas9 is going to cut the DNA, we know the base sequence either side of that, right? And so we make a piece of DNA, or when I say we, they make a piece of DNA that has these two homology arms, that is, stretches of DNA that are homologous to these sections here. But in the middle of it, the piece of DNA that we want to insert into the genome is added between those two homology arms. So what's going to happen if we step our way through this is, again, let's simplify the diagram so we're looking at it. This is exactly the same thing, except it's not a whole extra chromosome. It's just a little fragment that gets inserted into the cell together with the Cas9 enzyme that's got its single guide RNA in it. So the scientist is going to put in a Cas9 enzyme with single guide RNA and these pieces of DNA as well that the cell will use as a template to repair the break by the Cas9 enzyme.
Let's all go to the lobby. Let's all go to the lobby. Let's all go to the lobby to get ourselves a treat.
These are tiny molecular machines, and they are doing this inside your body right now. To understand why, we have to zoom out. Every day in an adult human body, 50 to 70 billion of your cells die. Either they're stressed or damaged or just old. But this is normal. In fact, it's called programmed cell death. But to make up for all these lost cells, right now, billions of your cells are dividing, essentially creating new cells. And that process of cell division, also called mitosis, well, it requires an army of tiny molecular machines. So let's take a closer look. DNA is a good place to start, the double helix molecule we always talk about. This is a scientifically accurate depiction of DNA created by Drew Barry at the Walter and Eliza Hall Institute of Medical Research. If you unwind the two strands, you can see that each has a sugar phosphate backbone connected to the sequence of nucleic acid base pairs, known by the letters A, T, G, and C. Now the strands run in opposite directions, which is important when you go to copy DNA. Copying DNA is one of the first steps in cell division. Here, the two strands of DNA are being unwound and separated by the tiny blue molecular machine called helicase. Helicase literally spins as fast as a jet engine. The strand of DNA on the right has its complementary strand assembled continuously, but the other strand is more complicated because it runs in the opposite direction. So it must be looped out with its complementary strand assembled in reverse, section by section. At the end of this process, you have two identical DNA molecules, each one a few centimeters long, but just a couple nanometers wide. So to prevent the DNA from becoming a tangled mess, it is wrapped around proteins called histones, forming a nucleosome. These nucleosomes are bundled together into a fiber known as chromatin, which is further looped and coiled to form a chromosome, one of the largest molecular structures in your body. You can actually see chromosomes under a microscope in dividing cells. Only then do they take on their characteristic shape. Otherwise, the DNA is more strewn inside the nucleus. The process of dividing a cell takes around an hour in mammals, so this footage is from a time-lapse. You can see how the chromosomes line up on the equator of the cell. Now, when everything is right, they are pulled apart into the two new daughter cells, each one containing an identical copy of DNA. Now, as simple as this looks, the process is incredibly complicated and requires even more fascinating molecular machines to accomplish it. So let's look at a single chromosome. One chromosome consists of two sausage-shaped chromatids, containing the identical copies of DNA made earlier. Each chromatid is attached to microtubule fibers, which guide and help align them in the correct position. The microtubules are connected to the chromatid at the kinetochore, here colored red. The kinetochore consists of hundreds of different proteins working together to achieve multiple objectives. In fact, it's one of the most sophisticated molecular mechanisms inside your body. The kinetochore is central to the successful separation of the chromatids. It creates a dynamic connection between the chromosome and the microtubules. For a reason no one's yet been able to figure out, the microtubules are constantly being built at one end and deconstructed at the other. While the chromosome is still getting ready, the kinetochore sends out a chemical stop signal to the rest of the cell, shown here by the red molecules, basically saying this chromosome is not yet ready to divide. The kinetochore also mechanically senses tension. When the tension is just right and the position and attachment are correct, all the proteins get ready, shown here by turning green. At this point, the stop signal broadcasting system is not switched off. Instead, it is literally carried away from the kinetochore down the microtubules by a dynein motor. That's the walking guy. This is really what it looks like. It has long legs so it can avoid obstacles and step over the kinesins, molecular motors that walk in the opposite direction.
Personally, I'm astounded by these tiny molecular machines, how they're able to routinely and faithfully execute their functions billions of times over inside your body at this exact instant. So I think it's pretty likely that in future we will be able to develop our own tiny molecular machines that will be able to repair our bodies better than they can repair themselves.
Humans use technology to store an incredible amount of data, and we may reach a limit soon if we don't find new technologies. Fortunately, DNA might just hold the answer to our data needs. Google processes 3.5 billion searches every day. Every minute, 4.3 million videos are watched on YouTube, and more than 156 million emails are sent. On average, 8,000 tweets are tweeted every second, and more than 350 million photos are uploaded to Facebook every single day. It's predicted that by 2025, this figure will rise to 107 trillion gigabytes. If we don't find better data storage technologies, we could be moving towards a massive bottleneck in our storage capabilities, which could stunt growth for businesses and innovations. An alternative to our current storage devices is DNA-based data storage. DNA has a lot of advantages when it comes to data storage. For example, it's ultra-compact and easy to replicate. A single gram of DNA can store up to 215 petabytes, which is 215 million gigabytes. The entire world's data could be stored in just 1 kilogram of the stuff. And you can store everything from text to images and videos in DNA. It can last hundreds of thousands of years and won't degrade over time like cassette tapes and CDs. In a recent breakthrough, Microsoft and researchers at the University of Washington developed the world's first DNA storage device that carries out the entire process automatically. Using the device, researchers encoded the word hello into DNA and converted it back to readable data. The device first converts the digital files into binary code consisting of ones and zeros, or bits. The system then uses a specialized software that encodes the bits into DNA sequences using strands of the DNA's 4-letter alphabet A, T, C, and G. The device then synthesizes the DNA and stores it as a liquid. Next, the stored DNA is read by a DNA sequencer. Finally, the decoding software translates the sequences back into bits that can be understood by computers. This DNA storage device may be amazing, but the concept is not brand new. Scientists have been storing digital data in DNA since 2012. They encoded a 52,000-word book and thousands of small pieces of DNA. But the method they used was inefficient and the storage capacity was very limited. Per gram of DNA, they could only store about 1.3 petabytes of data. Even though techniques are improving, before DNA data storage can be used commercially, the time and cost of synthesizing DNA and decoding the information needs to come down. It took a whopping 21 hours to write the 5-byte hello message and read it back out. The device alone costs roughly $10,000. Adding this to the cost of precursor materials to synthesize DNA makes the process uneconomical. But DNA technology and synthetic biology are moving at an unprecedented rate. It costs $2.7 billion in 15 years to sequence the first human genome. Today, all it takes is 2 days and $1,000 to sequence your entire genome. Everything alive demonstrates that DNA is already the world's most flexible and useful storage medium. We just need to learn how to harness what nature has been doing for billions of years and utilize it in our technologies.
The issue of embryonic stem cell research is surrounded with controversy. But much of that controversy is the result of misinformation. Dr. Sean Morrison helps us separate fact from fiction when it comes to the stem cell debate.
Stem cells are unspecialized cell types that have a remarkable capacity to give rise to large numbers of other specialized cell types. Embryonic stem cells are one type of stem cell that people are very excited about because these are cells that come from the very earliest stages of embryonic development, from only microscopically small clumps of cells. And these cells have the capacity to make every cell type in the body in unlimited quantities. And so when you're really trying to cure a major public health problem like juvenile diabetes or Parkinson's disease, the capacity of embryonic stem cells to make any cell type in unlimited quantities is an extraordinarily powerful advantage. The things that people should know to really make up their own mind about how they feel about embryonic stem cell research are that there are thousands of embryos that are routinely discarded by fertility clinics in this state and throughout the country. Those embryos, for a variety of reasons, could never be used for fertility treatment, but they could be used to derive new embryonic stem cell lines that represent the best hope for thousands of patients in our state and in the country. The state and federal restrictions that exist on embryonic stem cell research don't save a single embryo from destruction. These restrictions only delay medical research because the embryos that would be used for this research are already being thrown away. There are many restrictions that scientists observe in this area, and I think nobody feels more strongly than the scientists themselves that it's really necessary that this work go ahead according to the highest ethical standards. The bottom line is that if we really are serious about fighting disease, we should be using every weapon in our arsenal. We shouldn't be fighting disease with one arm tied behind our back. We should be pursuing all types of promising research so that we get to the cures sooner rather than later.
This raises a number of ethical questions that we have to carefully consider. And this is why I and my colleagues have called for a global pause in any clinical application of the CRISPR technology in human embryos to give us time to really consider all of the various implications of doing so. And actually, there's an important precedent for such a pause from the 1970s when scientists got together to call for a moratorium on the use of molecular cloning until the safety of that technology could be tested carefully and validated. So, genome-engineered humans are not with us yet, but this is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough. Thank you.